My accent is from the French-speaking part of Switzerland. And I really left my mountains because this university I thought was the most interdisciplinary I had seen, and that's really what decided me here to come. And today I would like to introduce you a bit to the exposome and really predictive modeling. I mean, I have a group of modelers where we try to go across different scales and from the environment up to inside our body. When I came here, my goal to enter a school of public health, I was coming from environmental engineering, is to have models which really go from world to centimeter in the body or even nanoscale in the body and do that. And that's what I pretended I would do when I was interviewed here, and Genomewide was in the committee, so I still remember him at the back of the room and thinking what he is thinking of what I'm promising. But what is interesting is we are doing it. I mean, nowadays we have projects which go, I mean, span the scale, literally from world scale to nanoscale. Now, to link it to more the domain here, we are in an era of high throughput measurements and high throughput biology and medicine, be it on the genome project and where we start having a lot of data arriving. The question is, what do we do with all this data and how we handle it here? Now, we have this Genomewide Association where we start to associate phenotype with genomes. I'm not a specialist of that. This is courtesy of Dr. Chirag Patel at the Harvard School of Public Health, who has, I mean, started to think of on the exposome and the link here with the phenotype here. So the idea is to say, how could we, in addition to the genome, which is one piece of information, the other thing which is influencing our phenotype is our history of exposure. And could we create an EVAS, which would be an exposome-wide association, in addition to the genome, complementary to the genome, and work that in conjunction together. So if we think of these two aspects together, I mean, the phenotype is influenced by our genome, but it's only part of the story. And we have this history and lifetime history of exposure. And basically, what Chirag is suggesting is, say, the variability of our phenotype between individual is in fact linked, and we have association up to 50%, but often less than that, with our genome. But the other 50%, in fact, are likely linked to the exposome. And that's what we would like to have. Now, what I would like to speak is how high throughput exposure data, not only genomic data, but exposure data and models can enable us to identify the role of the exposure in phenotype and capture this missing variability of the phenotype linked to our history of exposure. So, building on that, and I mean, Wild in 2005, I think. Here's the 2012 definition, but the first ID were arriving from 2005. But in 2012, there is this definition of the exposome. The exposome is composed of every exposure to which an individual is subjected from conception to death. And then there are more details. So it's really your history of exposure. Now, exposure to what? Exposure to chemicals is one thing, but it's also your food exposure, what you've been eating since birth, have you been breastfed, and there are all these elements which are influencing who we are presently, and looking at that all together. So my perspective on that, I would say, so far the exposome is rather a concept than an operational approach. Most of people working the exposome arrive from a biomarker perspective, which is one of the aspects here. The problem with the biomarker is you usually measure it now. You don't have longitudinal study, and that means that it's hard to know what have been the lifetime story of the exposure. And we know that, and the suggestion is, in fact, what's happening in our first 20 years, in fact, the first years, but the first 20 years are determining on a large share what's happening afterwards. So we would like to be able to go back in time. But the biomarkers, certain, if a substance is really persistent, this is bad for us, in fact, but perhaps we can reconstitute, and I will show you example of with dioxin, where we can reconstitute back probably from the present level how much people have been exposed in the past. But for many substances, we can't. And that's where we need modeling approach. So my perspective is to say, tell me when and where you lived and worked. Example, an Australian guy, he's been at school in Alice Spring. He studied perhaps in Adelaide, and he's been working then from 1999 to 2014 in Dow Corning industry near Sydney. Okay, so we should collect this information to say where people have lived, then what you have consumed and eaten. I mean, your food, what type of food you're eating. This is more difficult to get. I mean, the consumer product, we are using thousands of products nearly on a daily basis, but we can differentiate probably people who have a lot of, I mean, using a lot of cosmetic, people who are exposed to a lot of electronics, and which will affect this exposure and this history of exposure. Then we want to know the occupational exposure, what type of occupation you have at different periods of your life. And together of that, we could perhaps tell, and I will tell you who you are. Of course, that's your exposome, and that's only part of the story. But that's bringing that together. We could do that, and then we can start comparing with biomarkers data and see if our prediction are able to do that and start predicting and obtaining consistent results with the biomarkers. So, in fact, since I hadn't money to do that exactly, I say, okay, I was teaching last fall a class which is our capstone class, and there were 19 students. So I told them first we have 100 hours per student to invest in that course. So instead of doing a course, we'll do the Exposome 600 for project, and we'll try to do that for 10 different chemicals, okay, and see what we can get. And we got some interesting results. Some of them really liked it. Some of them hated it because they had to work hard. And the previous course was really easy, so it was interesting. So the long-term objective, and that's not only for that course, but our efforts on the modeling and prediction of lifetime exposure is to be able to trace and predict the lifetime exposure of individual to chemicals and also to nutrition on the basis of the time of evolution of their home location, economy sector, workplace, consumption habit, eating habits, and with, if possible, an emphasis on the early stage of life. Now, I find since I arrived here and was interviewed by Jill 10 years ago, or 11 years ago, in fact, we are really in an exciting time. Because as you, in the field of genomics, you have a lot of data arriving. In fact, we also have on the exposure side, a lot more data than even 3-4 years ago. If I give the example here on NHANES, we have with the survey which are done, we have a lot of data on the dietary intake, on demographic, occupational sector, but also about 212 chemicals measured in up to 47,000 people. And that, I mean, the main data are 99 to 2014, but we have some data earlier than that in 1985, 87. So we can start at, it's not the same people, that's the problem. It's always cross-sectional representative sample of the US population, but that's really useful. On the occupational health, we have some chemical exposure health databases where inspectors are measuring every time they go in an industry, and they report what are the measurements of about here 250 chemicals in about 900 industrial sectors. So we have this database which usually sit there and is not exploited, but it could be useful to look at to get an overview. Usually people working on occupational health would work one sector at a time. They will study one sector. But here we are interested to get an exposome for the entire population. So we would need such database. On the chemical, in consumer product databases, we start having database. This is a database from US EPA where we've been involved with them, how to structure it, to link it with exposure. We have about 500,000 product chemical combination available, saying which chemical are used in which product. And that starts to be really an interesting data. In addition to that, we start for certain class of chemical, detergent, cosmetics. We start having what is the typical fraction of chemical in the chemical content in the product as a function of the chemical inside the product. So if it's a surfactant, typically we'll have a certain range, whereas it's a fragrance in a shampoo that will be much smaller percentage than for a surfactant. On our end, this is our group. I'll come back on that. We are developing a multi-scale multimedia model, which go from about 10 kilometers to world scale, working with 120,000 cells, which enable us to know if you've lived somewhere and we have an emission inventory, we could start predicting what was your history of exposure to different chemicals. And then different data on the food and pesticide that we have. So how to use that? And I will walk you in the 4 domain that we had here. First on environmental, sorry. First on the environmental side, on the food side, consumer product, occupational, and come back and finish with the biomarker. So I will not go in depth in each of these domains because we need a week to look at that. But I will give you an overview of what, I mean, the type of things we can do, and then perhaps you will be interested to have some collaboration, I would say, based on this. So, as I was saying, when I arrived here at the lecture where Jill was there, I was promising, in fact, world to centimeter, and Martin Filber of DINT, who was working on nanotechnology, said, why don't you go to the nanoscale? And in fact, as I'm saying, I have presently PBPK model of nanoparticles, which are working at this scale. And this model, which is here, Pangea, it's a multi-scale model, which can go 10 kilometers to scale, and we can vary the resolution depending on the source. Here it's for benzene in Australia, emitted in Australia, and we had the sources. And you can see we have a high resolution where we have a lot of emission or high population. And we can start really covering, in fact, the entire world with this sort of model without making it exploding. I mean, you can still run it on a PC, in fact. A really fast PC, but correct. I mean, you have Alice Spring here, and that's about it. I mean, you have really not much going on here. The other domain where we have progressed a lot and with time is the near field exposure. We realize, in fact, that a large fraction of our exposure is indoor, and what's happening. I will come back on that. So if I take that and about, I mean, asking a person, where did you live? You can see that this is for benzene. The intake fraction, that's a fraction of the emission which is taken in by the population. So if I emit a kilogram of benzene in Alice Spring, here in the middle, I will have, in fact, a 2 parts per million. So that would be 2 milligrams taken in by the population living there in that case. It's interesting, if you have an offshore platform which is located here, close to Indonesia, in fact, the fraction taken in is higher than in Alice Spring. because you have billions of people. I mean, you have 100 million people living here in Indonesia, and in fact, they are downwind partly from there, so they get more exposed. And here you have nothing happening until basically Sydney and Melbourne, or very little population here. Now, if you emit at Orange Depot here, this is about 150 kilometers west from Sydney, and the dominant wind are this direction, then you start having 5 ppm, and at Sydney Airport you have 23 ppm. So we start being able really to predict what is the variation, and you can see how this intake fraction vary with distance. This is for the emission in this 150 kilometer west from Sydney, and you see the cumulative intake by the population, distance from the source, and that's different substances. So, for example, benzoyl is a very quickly degraded substance. It's only taken in very close to the source and doesn't increase afterwards. So it's really the population in Alice Spring, which is small, which is taken it in. But if you have benzene here, which is this curve, you see that you have a certain intake here in this region, and suddenly the pollutants is reaching Sydney, and you see that the large share of the intake will happen in Sydney. You have still a small bump here when it reaches Melbourne down here. Population, it increases a bit, but this is relatively small, and you get your total intake here of about 5 ppm, as I was saying before. So the message is we can start modeling for different regions of the world, and in fact we have data now of this intake fraction for more than 3,000 cities worldwide, and you can see here, I mean, if you're living in Lansing, it's only 5 ppm because there are a lot of countryside around. We have downwind from Ann Arbor, Detroit, larger population, so we get, in fact, a higher intake here. And New York would be 49 ppm, but the highest are like in India with 260 ppm with a lot more people around, or Tokyo. But Tokyo has, I mean, is on the border of the ocean and has wind apparently, so it's not super high compared to other places. So we see that's for the environmental part. So now, if I move closest to us, that's our food. What is our food exposure, our food exposome that we can do. So this is a domain where the global burden of disease is a study by Lim et al. 2012, which we are looking at and Murray from Harvard, looking at the risk factors of death worldwide. And the main risk factor, in fact, is associated with, I mean, food patterns and, I mean, deficiency in fruits and vegetables, or too much sodium and this sort of. Any idea in US how many people are dying because of bad food habits? Estimate per year? Any? Heart disease linked to that. It's mostly cardiovascular disease that are linked to that. It's 800,000 a year. Okay, so I mean, violence is sad with 30,000, which is far too high. But we can see that this is 800,000. I mean, it's huge. And in fact, it's not only US. It's even more pronounced in US. But nowadays, worldwide, this starts to be the first risk factor. Higher than tobacco smoke, in fact. And here it's clearly higher than tobacco smoke. The pollution is not negligible. Pollution in particulate matter kills in US 80,000 people a year. Okay, even with our relatively blue sky compared to Beijing. So think what's going on in Beijing, where you don't see the sun at this level. But the food is really number one, in fact, preoccupation. So we had studied previously what is the total impact here of pesticide use in Europe. And we were arriving with a 2.6 hour per person over lifetime, but with a factor of 400 uncertainty. Okay, it could be up to 2 months, but that's very less than the impact of fruit and vegetable, which is in years. Okay, typically like 3 years extension of your lifetime if you eat enough fruit and vegetables. Here we have the study look. I mean, you can see that the highest exposure, these are the kilo applied per year here. These are the years of life lost, or disability adjusted life years, so years of life lost per kilo applied here, which, I mean, look at the residue. This is the residue in the different products. So it's clear that the vegetables and the grape wines are the one which have, in fact, the highest impact because that's where we have the high residue. That's where we apply also at the latest date compared to wheat. If you apply a pesticide on wheat, it will be treated, it will be cooked, and so on. So it's really the fruit and vegetable. And it's in Europe, it's in Spain, España, France, Greece that you have the highest. Because why? Not because these countries are bad in terms of use of pesticide, but as the one producing fruit. So here in US, you would find California, Florida, I mean the high producing vegetable and fruit up there. But total is 2.6 hours, is relatively small here. Now, we've been working, I have a PhD presently working on this subject, and that's preliminary results here. But basically we have here, these are microdali per person and day, okay? And so it's 1 millionth of years of life lost. Okay, so it's about 30 seconds. Okay, so 30 seconds, and that is for 1 serving of fruit here. So that would be 161 grams of fruit consumed, or additional fruit consumed. And you saving daily about 50 microdali. That's about 25 minutes a day. That's, I mean, for eating your fruit. Saving 25 minutes of your life, that's high, in fact, when we look at it. The impact are here. These are, in fact, the particulate matter 2.5 impact related to that. That's linked mostly to NH3, I mean, ammonia emission, because you put fertilizer and you will get some ammonia emission, which are really orders of magnitude below. These are my pesticide here in green, but that's a factor of 400. Okay, but clearly we still uncertainty, we're still well below the benefit. So the message here is, in fact, in terms of exposome, I mean, eat the maximum fruit, and if you can afford the organic, afford it, but I mean, it's producing cheap fruit and vegetable that the maximum people, and good fruit or vegetables so that people eat them, tasty one, that we could move there. But we start having and we start a project now, in fact, in collaboration with the dairy industry, where we will compare, in fact, the entire diet of US at the US level, and start associating what are the risk factors associated to each diet. The third domain is the personal care product. As an environmental engineer, when I arrived here, I was working on environment, and I was working on life cycle assessment of products. Okay. And we were stupid because we worked a lot on the environment and we were neglecting the impact of the products in the near field environment. And in fact, what we are realizing presently, and there are nice publications, we've been partly part of it, but also from EPA at the Research Triangle Park in North Carolina, and showing that, in fact, in enhanced, when we look at the biomarker, the high exposure are all chemicals which are used in consumer products. So this is the easiest factor. Is this chemical used in consumer product? Is the best predictor to see the high biomarker usage of this chemical compared to chemicals just used in the industry and which are not transferred to the people. So now the challenge is, we have about, I mean, according probably at least 20,000 chemicals at high doses or high production volume, and up to 85,000 chemicals which are used in millions of products. So how to handle that and to do that? And I show that we start by domain, and here we have addressed the cosmetic, but we are working on detergent, we are working I have a postdoc presently working on chemical encapsulated in carpet, in flooring, or in different articles. And basically, we can have some archetype model that we can use and get prediction of what's going on. I give you the example of parabens. The parabens for 13 different personal care products that we have. Now, parabens are really, in particular methylparaben, are found in multiple personal care products. Nearly 50% of the product are using it. It's a preservative. And you can have different. You have methylparaben, butylparaben. You have different here, propylparaben and ethylparaben, which are here. Now, let's see to assess what would be the exposure. We need first, of course, to look how much people are consuming or using this product. And of course, everybody is in principle using shampoo. And if you have been using a shampoo today, you're very likely to still have some parabens in your blood just now. Okay. And we see, okay, I mean, there are variation between people, and that's also the variability. As we have the variability on the genotype, we have the variability on our exposome, where we have people putting about 3 times more shampoo than others, or taking more showers than others. You can see on body lotion or face cream. I mean, the people here are the one you were passing the street. You say, "Ooh, you cannot see the face anymore." Or, who is this person behind the makeup? And we see that. So that was interesting to see that. In fact, the shampoo, conditioners, body wash are the highest usage of personal care product, and the body lotion is also there. Now, we need to look, this is the application. Now, how much is going in our blood? And that's where we are developing, I would say, high throughput models, which can be used for thousands of chemicals relatively rapidly. And we've just submitted a paper with 700 cosmetic chemical in 13 cosmetics on that. And you have here the time, and this is in hours. So this is 6 minutes. Basically, here it's 4 minutes, and that's the average time people are keeping in US their shampoo on their head. Seems a bit long to me, but that's the reported average time here. And this is a body lotion where you leave on average like 8 hours on your body, or the whole day on your body. So you can see that in the environment, we were at 2 ppm, up to 20 ppm. So that was 20 parts per million, okay? When you put a chemical on your skin, it's, after 4 minutes, it's already the order of magnitude of 1%. And if you leave a body lotion, it's like 60%, which finish in your blood. It's not nasty stuff. I mean, a pesticide is meant to kill. Okay, not us, but insects or weeds, or so it's clear that you don't want to have more than traces. But here, when we put something on our skin, and especially in the body lotion case, in fact, we start having relatively high doses in our blood. And even if these are much less toxic material, the dose will be much higher than pesticide or this type of chemical in our blood. So in fact, what we are realizing that the shampoo, the conditioner, this is a log scale, it's multiplied by about 2%, and we are really dominated by the personal care products that are left on our body, and the body lotion is really the highest. So my recommendation is, since I did that study, I switched to for moisturizer and things like that. Coconut oil that you find in Kroger or whatever, you find them on the food. I mean, it's always cheaper to go on the food side than to go on the cosmetic side. And the only ingredient, if you look at the ingredient on a bottle of shampoo, you have about 20 ingredients. The only coconut oils, and as moisturizer, it works, at least for me, perfectly. I mean, So if you need some, you can, but that's very easy to avoid that. So now, is that real? I mean, that's our prediction. We're predicting blood concentration based multiplying this product intake fraction. It's a fraction. The previous graph was what fraction of, if I come back, what fraction here of my chemical applied in the shampoo finishes in the blood. And that was where we had this 60% here in that case. So if we look at the parabens, what is measured in the US population? And we see that first, even in the mean population, we don't need to have the high-end user. We are observing measurable and detectable values for this methyl paraben, which is the mostly used in that case. And we can measure the concentration in urine. So now if we look at the high-end user, the 95th percentile, yes? Yeah, so this is the 95th on the mean, huh? So that's not, if you like, it's the range of the mean, and this is the range of the 95th percentile. Okay, in term of accuracy. So first, we could expect that women and female have a higher urine concentration than male, but very interesting. Okay, you can see what is the trend, and you see that in fact women are about stable, whereas men between 2006 and 2010 have increased by nearly 50%, 40% increase. Okay, and when I see my sons who are 19 and 23, I'm not that surprised, because I mean, when I go on holidays, I take a small bag for my toothbrush and so on, and they always arrive with something, with a deodorant and I think I mean the advertisement have worked very well with the new generation of men also, I'm afraid. So I'm afraid they will be very soon at this level here. So now, this is what is measured. How does it compare with what we have predicted based on our use of product, the fraction of the chemical inside the product, and the product intake fraction, the fraction which goes in. And you can see we have here the measured urine concentration, and these are predicted urine concentration for the 4 parabens. And these are the different percentile of population. This is based on a stochastic model where we were looking at what is the distribution of application in the population. So I would say that's not high throughput. That was really a detailed study. But we can really reconstitute the variation within, I would say, a factor 5 of the measured concentration. So we can predict. So the model are not theoretical; they are relatively accurate at this level. So that's for the consumer. So the last really big part of the exposome is our occupational exposome. And we know that people working in chemical industry, I mean, we could expect that they are exposed at the higher level. How to address that? I mentioned a few minutes ago that we have concentration available, and here you have for that's in fact the 7 highest in terms of impact chemical used in the industry. And we were looking at the air exposure. So that doesn't account for dermal exposure or potential ingestion or like that. This is breathing, people breathing at their workplace. And this is a range of measured concentration of the different chemicals. You can see that styrene and dichloromethane here have the highest concentration, and a large variation, of course, across the different sectors of the industry that we have. Now, what we did is, now we've combined that with the number of hours. Here you have the percentage of hours worked in each sector. And here you have the concentration measured by the typical breathing rate. So you get the kilo per hour of the chemical that you would inhale per hour worked in this sector. That's what you are inhaling. And you can see we have, in fact, in 20% of the sector, we have about, in that case, 80% of the exposure is happening in a restricted number of sectors. So 20% of the industry are generating 80% of the impact at this level. Interestingly, it's not necessarily the highest sectors. Coating, engraving here, basic chemical manufacturing is still here. Household appliances, so it's more, and plastic product manufacturing. This is in fact the highest impact here for its formaldehyde, styrene, is in plastic product manufacturing. So it's not the one producing the styrene, it's the one using them in the product. That's where you have the highest exposure in that case. And so we have now across 250 chemical in 400 sectors. We can know if depending, you can say, I've worked in this industry, we can find the sector, and this is valid for blue collars. If you work in the offices of that sector, you're not necessarily exposed to that. So we differentiate between blue collars and white collars. So now, is that real? We look at NHANES and the survey of the biomarker, and for styrene, if I take this example, you can see the total NHANES and the non-blue collar here, and this is the blue collar industry. And nearly all high people are working as blue collars in the industry. These are really the high. If I take a log scale here, we see very clearly that on even on the level of the average, we have a higher blue collar industry compared to the non-blue collar, and we have especially the high-end people. This is a person who is working permanently in the industry on the line, what we expect they could have. But it's clear that in NHANES, they don't measure at the workplace, they measure people when they come back, and that's a few hours, and styrene disappears very quickly from the blood. So you would have a reduction of already orders of magnitude in what we are measuring. Now, if we look statistically, it was interesting for styrene, if we compare here the blue collars and the non-blue collars, we had, in fact, here a factor 17, I think, here. That's the highest. Factor 17 between the average of the blue collars and the average of the non-blue collar of the general population. So here we have the general population, here we have the blue collars working in industry. And we have here 30% increase, 20%, but you have to realize this 30% increase is in fact, most people in fact in the industry are even not exposed to that. It's a few industry where you have a lot. And that's what we are going to study and access. In fact, we need to have the confidential data from NHANES to apply for that, and you can then access the exact sector where they are working for. To finish with, this is really a study on the biomarkers. When I came here, I had colleagues who had the University of Michigan dioxin exposure study, David Garbrandt and colleagues, and they had measured, in fact, the blood concentration in 950 people of people working along the Titabassi River, which is a river which flows through Dow Chemical or along Dow Chemical in Midland, Michigan. And the question is, does the dioxin, because already in the 1920s, and most of the pollution has been there, also with Agent Orange during the Vietnam War, the production, there is a lot of furans and dioxin in the sediment of this river. What's happening every 3 years, it floods, it brings the sediment on the properties, and it means the soil are really highly loaded with furans. Question, does it go in the blood of people? Okay, and that's, we were wanted to reconstitute here the dioxin exposome of these people. If I go for that, we can see first that the older people, 80 years old versus 20 years old, the older people are clearly higher level of dioxin. And that's linked to two things. That's linked because dioxin is a success story, environmental speaking. Dow, in fact, discovered it in 1970. There were chicken dying in Midland, thousands of chicken, and they were wondering what's going on. And they discovered the molecule. It's not produced. It's a byproduct of chlorinated chemistry. And then we stopped the incinerator. We stopped emitting it. We filtered it. And nowadays we have in the environment level, which are a factor 20 lower than we had in 1980. Okay, even if it's persistent. Now the second thing is older people tend to be a bit fatter, have more fat, and so they degrade it much slower than younger people. And so that contributes to that. So the beauty of modeling is that you can start to reconstitute what is your history of exposure. We are measuring here, that was in 2005. We measured the blood concentration of this person. And what is very interesting is, even in 2005, you can see that about 2/3 of the concentration inside the blood is in fact due to emission which happened before 1980 here. So most of the concentration of this person is still linked to its exposure 25 years earlier in that case. And I mean, this is based on modeling, but we could start predicting across the entire measured population. And we made a pharmacokinetic-based analysis of a regression where we took the exposure variable, and like a bit in economics, you have an actual value, you have spending every year, and you calculate what is the spending in year 2005. And here I do the same. I have exposure every year, but I instead of using a discount rate, I'm using a degradation rate. And so the contribution of which was 25 years old is reduced, and I sum it up in 2005. And when we do that, we can reconstitute and see, for example, people, this is the rank of increasing predictive serum in the population, and we can see most of it is in blue, is contribution prior to 1983. So most of the high people are linked to their exposure before 1983. We have a bit in between up to 2000, and red is after 2000. So I say it's a success story. So now we can reconstitute in terms of exposure to know why are the people who have higher exposure because they were living in the region. Mostly it was the Midland 40 to 83. That's the number of years lived in the region. Okay, before 1983. This is what is important. How many years you've lived. Second factor, most important, was this here, the light blue, which is here, was the distance, number of years lived with less than 10 kilometers from the plant. Okay, so the distance from the incinerator was important. And then the fourth variable was, have you eaten fish from the Titabassi River and the Saginaw River, which is highly polluted? And fourth, have you eaten meat produced in the river? We had in this study, we had the Mr. Beef, and Mr. Beef was raising cattle in the river. He had about 10 times higher furan concentration than anybody else. So, in fact, the good news is for most people, what is in the soil doesn't finish in your blood unless you have a pathway which create that. And typically, if you start eating the turkey from the region or the deer liver, then you are creating this sort of pathway and you get a higher concentration. I would not still fish in that river, or I would go upstream from Dow Chemical, but these are these type of things. To finish, so I think we are at a stage, and that's why it's exciting, that we can start also predicting not only our genome that we measure, but having some idea of our overall prediction and interact with a lot of different chemical interacting. I like to finish and say a system is more than its component. And this is the chant show of the nightingale at midnight from Miro. Very nice picture. And a Swiss artist reproduced this picture with the same element. Okay, and that's what This Swiss artist got, okay, Ursus Veli 2002. These are exactly the same element than in the previous picture. You can recognize the different shapes that we have here. But it basically said that a system is much more than its individual components. Okay, and when we are working on genome and exposome, we need to understand the system as a whole. We need to understand the interaction, and we need to identify the system and interaction, identify the role of each component, and in our case, understand the interrelation between the genome, the exposure, and the system behavior. Thank you for your attention. Questions? I don't know. Who dare to start?
So I
Oh, so- Are you also
planning to use some like a mobile, like the smartphone, and so how that information can be incorporated and then construct its lifetime exposure.
Yeah, I mean that's another possibility to say. I mean everybody nowadays have one of these things in your pocket, and I mean if we could have some sensors, we could directly measure the substances, but we can start measuring the location where people are. Now, do we want that to be followed? I mean, one thing or not, we are followed by Apple, and they know exactly where we are at which point. This sort of data could be very useful. The other type of data we are relying on here are, in fact, the EPA database, where I was saying on consumer product. A large part come from Walmart. Okay, and because Walmart has, for all its product, has the composition of chemical, so you can start having that, and you can start knowing if you make an agreement with a company like that, you can know the consumption habit and who is getting which product together. So exactly, I mean, from this sort of data, we start having a lot of information. Not always easy to dig. I mean, the tweets could be interesting as source, but it is, I mean, it's hard to validate and really do that. But typically, we are going in that direction to have. My dream is, in a sense, to people could have a small app and say, okay, what am I consuming a lot of that and so on, and then they get sort of picture of the exposome, where they work, which sector of the industry they've been working on, or I mean, which type of industry, how long in the year they've been working, and then we could reconstitute a sort of app doing that.
Some of the research on exposure to different substances has led to understanding of combinatorial interaction. For instance, acetaminophen and alcohol have some negative effects together that are not the case individually. How trackable do you think this kind of databases and approaches are going to be toward investigating these combinatoric exposures? Do you think there's potential there, or does the statistical power and the computability go away once you start investigating?
Yeah, it's I would say, regarding the number of products we are exposed and all that, it's hard. I mean, presently, I would say I don't start with interaction. I mean, I start what we can do at this stage. But it's interesting on the dioxin, for example, the smokers have a smaller dioxin concentration in their blood because they already activate their defense to try to get rid of the other substances. It's not because they have, I mean, but they degrade quicker because their immune system is working full time to try to get rid of the nicotine and other products they are bringing. It's well known for this interaction with smokers. So that was one factors where we used to predict. In fact, the half-life is lower in the body, is lower for smoker than non-smoker. And we have that. I think my position a bit with interaction is, let's start. I mean, we are exposed to a lot of things, so let's start to get without interaction. And as we are getting the data, we can start studying this sort of thing when we know it's important.
A second question. This kind of systematic, quantitative investigation of risks and benefits from various exposures is perhaps a useful corrective to some of the tendency that the press has to inflate findings. really interesting study that showed that of people who lived near the Fukushima nuclear disaster, among the few people who had higher ethane exposure than the rest, the bulk of it was coming from local mushrooms that they were cooking and eating, and immediately inflated it toward the notion that produce grown in Japan were the best. Do you think that there's any way to get this kind of quantitative evaluation of exposure risk out there into the test in a meaningful way, an encyclopedia of risk exposure, something like that?
That would be the sense for an app or something like that to be able to say what is the magnitude of my exposure and exactly to start. And that's also a bit the sense of this pesticide study, that in fact we speak a lot of pesticide. That's not the top priority. I mean, it's not super important in Europe and here because we've got rid of the bad pesticide. So it's because, I mean, and I'm still, before eating a fruit, I will still carefully wash it and try to reduce the maximum we can. But the priority is clearly, for example, in that case, to eat fruit and vegetables. And it's true that, I mean, it's a problem with the news that sometimes they like to inflate. And as soon, like, when some ideas have gone through, it's very hard to correct if they are not correct on the long term. But I mean, that's our job, I mean, to bring science-based evidence. And that's, I find why I've been hired here, that we provide that. And with time, I think messages can go through on the long term, not only the sensational one. Jill? Yeah.
Thanks very much. Enjoyed your lecture. You know, when you mentioned about washing the fruit, I have this image of my father when he was about 80 years old, washing the fruit. But first he put a filter on the kitchen spout so he shouldn't contaminate the fruit from the water. Of course, the chance it was going to do him harm in the short amount of months or weeks left was another parameter. So did you say that nicotine and other interacting chemicals were eliminated by immune mechanism?
No.
I think you mean by detoxification.
Yeah, that's correct. That's the. But if you like, the nicotine would start triggering your detoxification system and enhance, in fact, the degradation. I mean, your liver functioning with P4, the different enzyme would be at higher concentration because they're fighting something else, if you like. So in that case, the dioxin, I mean, it would also contribute to detoxify the dioxin.
So people might like to know that we have a center here in the School of Public Health plus Medicine on MLEAD. So it's Michigan Early Life Exposures with Late Adult Disease Risks. Mm-hmm. So in the beginnings of that, we assumed that we would soon find by epigenetics biomarkers for common metal exposures and other exposure of the kind you documented here. Do you, in any of the biomarkers you use epigenetic parameters? And are there any that are really specific for parts of the exposome?
Yeah. The answer is presently not, I mean, it's not my expertise, but I mean, we discussed with Dana, Demiloy, and together to see how we could do that. I think it's interesting to combine. The thing is to bring us together in project exactly that which is a bit this sense. I would say within that project, we've not been, I mean, I'm part of still of the exposure side. I'm not called in a sense. I mean, we are a bit on call available to do that. It would be interesting to start combining.
Okay, well, this is worth everybody's knowing about. The center is just, not quite officially, but just about been renewed by NIEHS, which is a big success. And they have interesting seminars. They have these core laboratories, such as the exposure lab that you are associated with. And there are good opportunities for pilot projects and other collaborations that I think some of our people might like to pursue. The lead contact really for us is Maureen Sartor, who heads the Bioinformatics core. I'm involved also with the faculty career development. So it's good. As far as I'm aware, for lead and other heavy metals and for various of these organic chemicals, we really don't have many examples of distinctive epigenetic, either methylation or histone molecular phenotypes.
To detect that.
Yeah.
So that's where I think like it's interesting, but very few people are approaching the exposome from a predictive perspective, and I think there is really potential in several domains to bring also this without necessarily being on the biomarker side, and complementary to the biomarker, we're able to predict. So I think there is a potential to do that, which is neat. Okay. Any other questions? Time to, I don't know.
Thank you very much.
Thank you very much. Yeah, you can think, and I mean, in fact, I'm here.